In this video, we will explain the process of high throughput screening considering the types of libraries available and where the products come from. Let's talk about high throughput screening or HTS. It's basically a way for scientists to test huge numbers of compounds, sometimes millions, to see if any of them have the biological activity they're looking for, usually against a specific target like a protein, enzyme, or even a whole cell type. The first step is picking that target and then figuring out an assay, which is just a method to measure whether something is happening with your target when you add a compound. A big part of HTS is the compound libraries. Think of these as giant collections of molecules all ready to be tested. There are a few main types. First, you've got natural product libraries. These are full of chemicals that come straight from nature. Plants, microbes, marine creatures, you name it. Natural products are famous for their complex structures and surprising bioactivities, so they're great for discovering new leads that nobody's thought of before. Next up are synthetic libraries. These molecules are made in the lab, often using combinatorial chemistry, which is a way to mix and match building blocks to create tons of different structures. The goal here is diversity. If you cover lots of chemical space, you have a better shot at finding something that works. Then there are fragment libraries. These contain small molecular fragments, usually with lower molecular weights than typical drug-like compounds. Fragments might not bind very tightly to the target, but they can be great starting points because they're easier to tweak and build upon. Finally, there are peptide libraries. Peptides are short chains of amino acids, and they can be either made synthetically or derived from biological sources. These libraries are especially useful when you're trying to disrupt protein-protein interactions, which are notoriously tricky targets. So where do all these compounds come from? Natural products are extracted and purified from living organisms. Synthetic compounds are put together in chemical labs. Fragments are designed to be small and modifiable, while peptides can be made chemically or produced using recombinant methods. The actual screening process is super automated. Robots dispense tiny amounts of each compound into microtiter plates, and sensitive detectors measure the results. Researchers look for hits, compounds that show the desired effect on the target. Once they find hits, those get validated and optimized through further testing and tweaking. Eventually, this leads to promising candidates for drug development or other scientific applications. It's a fast, efficient way to sift through mountains of possibilities and zero in on the ones that really matter. See you in the next video.